Bienvenidos al Seminario Fronteras de la Energía, organizado por el Instituto de Energías Renovables de la UNAM. Antes de comenzar, pedimos a los estudiantes de la Licenciatura en Energías Renovables que agreguen las siglas LIER a su nombre de usuario para contabilizar su asistencia. En esta ocasión tenemos al profesor Piet de la Universidad de Galway en Irlanda, y por cortesía de nuestro invitado, voy a cambiar al idioma inglés. So we're glad to have Professor Piet here today with us in the seminar. I'm going to do a brief summary of the academic information of Professor Lenz. Professor Piet Lenz is Professor of Environmental Biotechnology at the University of Galway in Ireland. His research focuses on biofilm, sulfur biotechnology, metal speciation, bioavailability and removal, natural treatment systems, anaerobic wastewater, and waste gas treatment for resource recovery and reuse. He is the initiator of the Marie Curie training site Sulfur and Metals, the Erasmus Mundus joint doctoral program, Environmental Technologies for Contaminated Soils, Sediments, and Solid Waste, among others. He's founding editor-in-chief of the review journal Reviews in Environmental Science and Biotechnology, and founding editor of IWA publishing series Integrated Environmental Technology. He has co-authored over 700 scientific publications and edited 15 books volumes. Besides innovative research, he is also a leader in education and capacity building, organizing numerous study days, conferences, summer schools, and short courses. Professor Lenz, thank you very much for accepting the invitation to give this talk. You may start, be my guest. Thank you.
Okay, thank you very much for the invitation to give this presentation. And it's a pleasure to discuss with you some work that we have been doing on the anaerobic or on the recovery of rare earth elements in different reactor types, mainly with anaerobic microorganisms. I will also present something about the use of fungal bioreactors for recovery of metals. So in our group, we work on reuse technology, resource recovery for organic and inorganic waste streams. And for today, I will give a brief introduction on the topic. Then I will talk about specific metals elements. This is from zinc, purification residues. Indium is also interesting, and then rare earth elements. So yeah, more and more we have next to the biofuels and the biological production from substitutes of organic fuels like diesel or gasoline. We also have the green technologies like wind energy and solar energy. For that, we shift actually our needs from organic materials to inorganic materials, and there these elements that are mentioned here, they play important roles in order to have these techniques working. So I will be giving examples on how we can take biological processes, so with microorganisms, in order to recover these elements. So the introduction is that at the moment, I think, all over the world, we see, quite considerable issues, problems, and so there are next to, where we started from, at least where I started from, natural resources and also the environment. And there are also, nowadays, we have also a big attention to climate, to health, to livelihoods, the environment. So it has become more complex. More factors have to be taken into account when we are developing solutions. And still this slide is the last year I can show it. I'm showing it already several years in the lecture. So we are now 25. And so it's very important to realize the population growth. So I think we have achieved already a lot, actually, clinically and also as economies over the last 20 years, about the last 50 years. But yeah, the fact that we have this huge population growth, especially in Asia and Africa, it means also that we have increased pressures on food, fresh water, but also space environment, natural resources. So the question is, can we keep the pace of serving all the people that are now being born? Can we establish the same or maintain the same conditions as we have had in the past 20 to 50 years? And that's a big challenge. And their reuse technology become very important. And actually, this makes us now very, makes it more acceptable that we talk about element reuse, mineral reuse, water reuse, food scarcity. So what to do with food waste, energy scarcity. So the big challenge that we have is if we talk about energy, like our institute, Instituto de Energías, Renovables, but it is not only about this energy. There are interlinks with minerals, as I was just saying. The wind and solar panels are very much depending on elements, but also we need water for mining. And yeah, if we have the water and the minerals for energy and water, what about food? So it is quite complicated. And how do we make a balance between these four factors? It will be determining in the next few years if we can still maintain the quality of life as we have it now, with all this extra people still coming because of the population growth. So to solve today's societal problems, it is helping us actually to push through the circular economy. So 20 years ago, mainly we had the emphasis on cleaning the environment, but now we have connected this to biodiversity, supply of raw materials, also climate change. So these drivers help us in some way in order to create technologies and supply chains so that the circular economy can become a reality, and so that we don't need to mine for this new minerals or water, so that we can reuse. This is being supported by our policies in order to cope with the issues that we have. And the net zero transition strategy of Taiwan, they are taking this very serious. So they want to have the whole Taiwan energy neutral and carbon neutral by 2050, so 25 years left. And they have made many different items. They have identified there is a lot of renewable energy there that is for where we work on carbon free vehicles, but also here resource recovery and so on. So there's a lot of scope for the work that we are doing in our centers. I would like to emphasize or to draw your attention that next to the energy and the industrial transition, so next to technology, this Taiwanese program, it also has the attention to lifestyle transition and social transition. So this is the first time that I saw that next to technical interferences or transitions, there is also an attention to lifestyle transition and social transition. So Taiwan is a more, it's a country with more guided society, to say. So there's more top-down approach. So this lifestyle and social transition can be made there more easily. But I think that's very important that next to how we adapt our processes, that there is also attention. I think one slide, attention is being given to social and lifestyle transition. That's I think very interesting and also very much needed in order to have this transition possible. We are more in the technical part, and so today we talk about, can we substitute, so next to the new resources is here, and the mining, can we take substitute materials in order to feed in the processes. Other processes or other research that we are doing, and I'm sure also you in Mexico are doing there. So can we maybe instead, so we have here the basic industry, the development industry, and the users. This is the waste, this is recycling. So can we narrow the loop? Can we make these loops smaller so that we need less materials? And also, can we close the loop? So this is really reuse, the circular economy. So different interferences at different places, we can look and make adjustments in the processes in order to have less materials, less new materials, and that we can here the waste processing, that is less, and that we can reuse more, so that we need less primary resources for the whole process, as we have been doing that. And we are working on this, and I will show you now the rest of the presentation, these examples from steam processing industry, lead industry, indium and rare earths, where we have been looking, can we develop these reuse processes, and can we use bacteria, microorganisms, so environmental biotechnology for these, for the recovery of these elements. So germanium and helium are the so-called critical elements. They are used for different applications, also among others in solar panels, in this wind turbines, in renewable energy, but also in smartphones, electronic industry. So there is a big interest in that. Also here, rare earth elements, we'll speak about that in the last part of the presentation. It are highly important elements for the economy, but the supply risk is at hand. And I think yesterday or day before yesterday, there was actually also an indication that US, with the new government, they are trying to block or to also work on this rare earth supply to make changes there, so that they are less dependent of China for that, because at the moment China has this dependency on many of these elements, and also South Africa, by the way. Many of this, as you see here, China, I'm not sure if this is for you, blocks, but China, this is 2020, provides a lot of these elements. Mexico, actually, also some of them not neglectable. A mining sector in Mexico is also not neglectable, but compared to that, what we take from China, this is huge. So these recycling, reuse technologies allow us to to become less dependent of this monopoly of China in some way. So in our philosophy, the way we approach it, we look at the waste materials, pollution, and then with biological processes here, it is depicted as a USB reactor. We are looking, can we, with these microorganisms, produce useful nanomaterials that can be either functional. There are examples that, for instance, cadmium sulfide can be used as a nanomaterial in a diode, so in electronic industry. Here, cadmium telluride, some type of solar cell is using this. And here you see one microorganism, Pseudomonas. It's an aerobic one. This is how you grow it in a normal culture. And then here we see the red, the biogenic red elemental selenium. If we take a picture of that, so this is a bacterium, 1 micrometer. So this is a Pseudomonas bacterium. And then when we expose it to selenate, the microorganisms produce either elemental selenium, or and heavy metals are present, also cadmium selenide or zinc selenide are produced, and these find an application in some of the electronic industry are. We can use them. We have been looking a lot for, can we use these as absorbents for heavy metals, among others, the ones I will talk about now, the gallium and the germanium and zinc. So this is a first study from PhD students, where we looked at the elemental sulfur, selenium, and tellurium, either chemically produced or biologically produced with the microorganisms. When we give it then tellurium, they form this black suspension where elemental tellurium is formed. Selenium is red, and sulfur, well known from the desulfurization of biogas, it has a yellow color. So we combined that, and well, just to, this is some background on how we do that. This is anaerobic granular sludge. So it is anaerobic microorganisms. We put them in a medium, and then we expose those to the metals, and then we look at how much metal has been removed. And then we are looking if there is nanoparticles, is the same. So the microorganisms can produce these nanoparticles, and then we look at biosorption by these nanoparticles. So this has been published in the paper. I will provide you the slides, sorry, for that. And so we look at, also in this study, we compared absorption to this elemental nanoparticles with sulfidic precipitation, also chemically produced sulfide. So this is just chemical sulfide, or biologically produced sulfide from biological sulfate production reactors. So the details you can find in that paper. And as you see here, we have here a notice that, well, here nothing is removed. Compared to here, we see some deviations. So the conclusion, so we can talk much more about that, but as you see here, so for zinc, gallium, germanium, for sulfidic precipitation, there is not so much scope, and the efficiency is not very good. But when we use biologically produced sulfide, then we can produce a lot, precipitate a lot, half, so half of the zinc and half of the gallium is precipitated, whereas germanium is hardly precipitating with sulfide. So it means that if we use biologically produced sulfide, we can selectively remove germanium from zinc and gallium because it is not precipitating, and then it can go to a next step. Similarly, we can look at the absorption of these elements to this elemental forms, and then again, sorry, we see here again. I just take this picture here because it's so easy to read. So we see here that the percent removal of gallium is very good, whereas again germanium is not removed, and now also zinc is not removed. So if we now take this waste stream from zinc purification residues, we can first remove germanium because it does not precipitate, and then the waste stream goes here, or we do the other way around. We do first gallium removal, and so with an absorption process, we can remove mainly gallium from the stream. And then with the precipitation, we can then remove the zinc from the stream. So like that, we can kind of chop off the different elements from biologically produced, from the zinc purification residue. So in these papers, we have then also been looking at, is it now ion exchange, is it absorption, is it precipitation? So there's a lot of there are microorganisms, so absorption on biomass. There are a lot of possible mechanisms, and then in the paper we have also been trying to allocate where we can find or what would be the mechanism of this removal. When we have the absorption compounds, we want to remove them, so we want to take them out as from this absorbent, and then we have been looking at, can we take oxalic acid or malic acid or acetic acid, EDTA, what would be the best? And a bit surprising, EDTA is not so efficient, but then these organic acids here, oxalic acid, everything can be removed. It is still quite high concentration. So, but also if once we have the metals absorbed, we can also desorb them effectively, and then we have a concentrated solution of this gallium that can be used further or reused then in production processes. So this is what I was saying, and again we have here published this information, and then we have also looked at can we go for column experiments. So here you see a column, so continuous flow experiment where we were looking at, so this is the beads. We immobilized nanoparticles in these beads. It is alginate beads, and then we packed those columns either non-supplied, just alginate, then alginate with selenium nanoparticles, alginate with tellurium nanoparticles, and then again we have been making characterization where we then also have opportunities to remove zinc, and so we see the different amounts of zinc that can be, of these elements that can be removed. We see that, so here we have 42. So all values are improved. So when we add these nanoparticles produced by microorganisms, we can also increase the removal, the amount that has been removed via these in these columns. So just want to finalize this with telling that we have a scope for removing, separating in zinc purification residues, gallium from zinc from germanium, and that we can, we have characterized, for instance, with immobilizing this nanoparticles alginate beads, we can go for a continuous flow process in absorption columns. So this was the first case study for the zinc, aluminum, and germanium. Then we can next that I will talk about is indium. So indium, I will talk about fungal bioreactors and USB reactors. So here this was absorption and precipitation with a biosorbent produced from selenium and tellurium. Now we will look at different process, a bioreactor, two types of bioreactors, one with fungi and one with anaerobic bacteria. So why are we interested in indium? It was kind of, our aim was again in this, here we see semiconductor industry. It has low melting points. There are specific applications. And again, in this push to, can we reuse these elements? We have been working, or we thought that can we also not consider gallium? So I showed you indium. So I showed you there this the results with for gallium. Indium can behave similar as gallium. So we were looking, can we also remove that because it has no biological role? We need to investigate, does it now work as well as gallium or not? And here for these users, there are the semiconductor compounds, also in photovoltaics, so also this famous theory with nanoparticles. If you give different elements, if you have nanoparticles of different sizes, we get a different color. So from that point, we are interested in recovering indium, and with this chalcogen, so with sulfur, selenium, tellurium, it makes this type of compound with various properties. So for this study, we used Aspergillus niger with our fungal reactors, and we were interested, can we use that? So before we worked a lot with phanerogator, and both Aspergillus and phanerogator, they are interesting because they produce this out of the spores. Out of the spores, they form pellets, and these pellets grow, they can break. But yeah, as we if we make the reactor operation right, we can maintain in this pellicle form, and we can avoid that this is at a given moment they will break down, but we can work like a USB reactor. We can work with this pellet. So this pellets, it is not, so here I showed you the alginate beads. So these are beads of alginate. We produce those like agar. It's agar, it are droplets. We can make a droplet of agar in water, and then you get these nice beads. Here we produce the beads ourselves with inoculating the spores of the fungus, in this case Aspergillus, and with a shaking, if you grow them in a shaking environment, we can make these nice spherical spheres that can also remove, that are remaining from, that can be retained in the reactor. So we talk about biomass retention. That for continuous reactors is very interesting. So here you see the morphology of this fungus, which are high-fi, that are compacted in a sphere. When we expose them to zinc, also selenium and tellurium, again we get these nanoparticles. There is no, so this is about 50 to 200 nanometers. So there are nice spheres. In other cases, we get this needle shape, so nanowires. We have also been looking at what can we maybe do other applications, but here we were now interested in if we expose both indium and selenium and tellurium, can we maybe get this indium selenide, indium telluride, or what happens when we give them both. So this is typically we take the fungus from a plate with the spores, we inoculate them 120 rotations per minute, 30 degrees. With shaking, we get a nice sphere. So here you see an incubation with 15, about 15 milligram per liter of indium, and then independent of the presence of selenium, we get the reduction of the indium concentration. It takes quite a while, so we talk about 10-15 days. So this is not nothing, and also it is a little bit strange that we stop here at 5-6 milligram per liter. So we have a removal of indium, but it does not go. It is not a complete removal. We think, it's likely this is due to the carbon source that is not completed. That is completely depleted. But you see here, when we start with 10, we get we go to 5, and if we go for 2, we have hardly any removal. So the initial indium concentration plays a role in how good we can remove the indium. We were happy we could remove this indium. Is it now? Oh yeah, here you see the color of. So this is this single bead with. This is the control. So we see these nice spheres, and then there is some outgrowth. If there is selenium and tellurium, you don't see this outgrowth. So selenium and tellurium are kind of have a toxic effect, and they are inducing better spheres compared to the control. If we have then indium present, it is not so much affected. And then if there is indium and selenium or tellurium present, we get mixtures in the colors. So I have shown that we can remove this the indium with the fungal pellets, but at higher concentrations the removal is better. If there is selenium and tellurium, better, especially tellurium was giving better removal efficiencies compared to the control where there is no tellurium. So we have a positive effect of removing indium with tellurium present, and then on the other hand, also when we look at the selenium or the tellurium removal, the presence of indium was also affecting it. The morphology was changing, but we were not really able to show. We have done XRD, so we could not show this indium selenide and indium telluride. So probably if they are formed, the concentrations were too low. So we have done the same with USB reactors with similar results. So here you see, if you want to have more information, you can check this publication here, where all this data are presented and elaborated. And then we have another paper on the USB reactors. But because I have taken this now out because of the time for this lecture. But also with for indium, we can also remove with USB reactors. And another item to point out, is that so there's a problem or a point to consider is to create what is not. Let me see. Yeah, here. So maybe you have spotted this or not. But 10 grams per liter glucose is really huge. So this is an issue. So how can we, so it means we need waste streams that have this glucose in order to be able to make this process economically viable, because we're not going to run reactors with 10 grams per liter. And also one other paper we have shown that if we substitute glucose with the leachate of a brewery spent grain, we can also make use of this fungal pellet. So we can substitute this glucose with acid leachate from, for instance, brewery spent grain, but could be any lignocellulitic material. So if the processes that we use to pre-treat the glucose, the lignocellulitic materials for acidic fermentation or for biogas production to unlock the sugars, we can take that sugar-rich fraction and use it to grow for the fungal pellets. So the last point or element group that I want to discuss is about rare earth elements. So there we have looked at absorption, and again, here we have the USB reactors. So these rare earth elements are used in solar panels and windmills. So like windmills, they have a lot of neodymium. And in this solar panels, we have a lot of lanthanum, neodymium, and also dysprosium. So we were interested, can we use these? If we would need to reuse, can we do acid leaching of these elements and recover them? So I already pointed out to the importance of China as supplier for these rare earth elements, and this 2020. I'm not sure how is the situation for the moment, but still this is a huge supplier of these rare earths, and as Europe, we want to become independent of this supply, and many of these elements. So here neodymium, also gadolinium, neodymium again in magnets, lanthanum in metal alloys, neodymium again neodymium lanthanum for catalyzers. So there is a big interest for recovering them, because catalyst chemical processes, we also need these or use them in biorefineries. So we need to look for alternative sources for these rare earth elements, and we look now at, can we recover this rare earths with are there other ways? So we looked at I will present you two ways that we can do that. The first one is sorption, and the second one is then removal in USB reactors. For the sorption, I need to take a side step because we took the waste biomass of biorefinery process. So in Ireland, there is a lot of ulva, you see here at the coast. Sometimes you have, so this is ulva, how it looks like in a normal situation. Especially in summer, there can be a lot of blooms of this ulva, and we are interested in that because this ulva, it's a green algae, they have the special component, ulvan. It is a sulfate-rich sugar that is used in medical applications. So we were looking at, can we maybe take that these sugars out, and what to do then with the waste biomass. So we took the ulva. Indeed, we can do a simple citric acid extraction, and then we have here the sulfonated polysaccharide. It can go for medical applications, or we can consider it for making it as a sugar for the drinking, as a substitute for glucose in coffee. But here we have looked at can we make a plasticization of this ulvan, and indeed with xylitol we can make a coating that we can spray on the fruit. So we have a kind of a plastic coating that is edible. It is an ulvan film, no problem. There is nothing, no problem to eat that. And so we have here then a edible plastic. The remaining biomass, we have done a lot of work on what to do with that. We looked at dark fermentation, we looked at biogas formation, we looked at what can we do with the CO2-rich biogas, but then with the VFA-rich element digestate, we have looked at the liquid fraction. We have then shown that we can, it's very normal, and with the VFA you can make this polyhydroxyalkanoates, and then we can make a non-edible film of plastics for the, for instance, for packaging of fruits. All information is here in this paper. The question is still after this, after dark fermentation, after we have the liquid digestate, we can make the plastics. What about the solid digestate? But there we thought, well, this solid digestate, or after extraction, the ulva-free biomass, we looked, can we, like alginate, it is very good for heavy metal adsorption. So, and we looked at, we have here then in this paper, we compared how can we, what about cross-linked ulva and whole biomass ulva, HCl existed, extracted ulvan extracted, and we looked at can we use this for adsorption of these rare earth elements. And indeed, we have very good removal efficiency. So if you look in these papers, I don't know if you have the values here, no? So, but here, especially in this biomass type, we can actually recover all these rare earth elements, and we only did these three. So we could also consider absorption of other heavy metals with this biomass. And again, then we have the question, what do you do with this metal-loaded biomass? Then we desorb it and then try to separate the different rare earth elements so that we can make use of them in the different applications. Then last process I will discuss with you is the anaerobic granular sludge. It is comes from USB reactors where we treat heavy loaded wastewaters with granules. Let me see if I have a picture of those granules. Here you see they are normally in water. So here you see these are small bulleted. They are like the single pellets. I'm sorry, it is not so well visible. But we use then those anaerobic granules to reduce selenate or telluride to elemental tellurium. So those can be a source of, again, these nanoparticles to precipitate out the rare earth elements, neodymium, lanthanum, dysprosium, or it can be precipitating directly. Maybe we don't need this elemental nanoparticles. We can just have the microorganisms that absorb or precipitate those. So we made a long test with many controls with these rare earths, with and without the selenium tellurium. We worked with lactate in this case, and we could show. So this has been published. I'm sorry, there is not a reference, but also here you can find back the details of this study. Also an abiotic test, we looked at different pHs, and that became a very important point, because here we were the first test, we were very happy because we saw almost 100% removal. So this said, okay, that's very good. We go for USB granules, they are perfect. But then we saw the results of the abiotic test, so no granules, and we saw that supposed if there is no biomass, it should be there, no change, but we also saw there that they were actually precipitating. So we realized that actually at the pH where a USB reactor operates at 6-7, there all the heavy metals or the rare earths have precipitated already, and it is actually a chemical precipitation and not a biological activity. So we have then tried, can we operate a reactor at 4, but that's a big challenge. But at the end we succeeded, and then we showed that there is some removal. So here you see the USB reactor. So this is one reactor. It is an upflow reactor. So the wastewater goes in here. This is the sludge bed, and then here we have the cleaned water. And we were looking in this sludge bed, can we there then remove the rare earths, and we still have to recover them from there. And then we had two. Oh yeah, here you still see the scheme. So we have the influent goes up the sludge bed. Here we have methane gas from the organics, and then here we have the effluent where the water, the clean water goes out, and then we have also a recirculation. And we had two, again, we checked for this, can we reinforce the removal of the selenium and tellurium? And just to go through it, we saw that at this time we finally figured out, yeah, okay, we have to operate at this low pH, because otherwise we don't enter any rare earths. And then we see here there is a good removal, but then suddenly there is a breakout of the heavy metal of these rare earths. So there is a wash out, is the desorption from the sludge bed. So we were actually not really well able to have a continuous good removal. So it's well possible, but we have to look at, yeah, can we need to do more research for the optimization of this USB reactor. So I think we can state that we are able to remove rare earths in USB reactors. We show the proof of principle, but we need more work to optimize those reactors and to show a more stable removal for a longer time. We also looked at, can we maybe make this metal or rare earth selenides or tellurides for direct applications, but it was also mainly dominated by the desorption of the metals. So from the batch assays, we were able to tell that these rare earths are not inhibitory, and that the presence of the rare earths enhanced the chalcogen removal. That's also very interesting. And in those tests at pH 7, it was mainly chemical precipitation, whereas in the USB reactors, a neutral pH favors the precipitation, again, mainly chemically, but then if we go to lower pHs, pH 4, we can achieve a very good efficiency. removal efficiency with concentration of 50 milligram per liter, which is not neglectable. So I think, as I said, we showed here the proof of concept, but we need to do further research or work at the conditions better to optimize the conditions for continuous removal. So with this, I come to the conclusions of this presentation. So the rare earths and also all metals that are important for the circular economy, we have a scope for biological processes for removing them. So not only the base metals that are quite well studied, zinc, cadmium, copper, lead. There we have a lot of experience. We showed environmental biotechnology can play a role there, but also for the less evident elements, these rare earths, indium, also platinum group elements, actually sulfate or biological processes can play a role. And which processes? Well, it can be sulfate reduction, sulfidic precipitation. It can be selenide and tellurium precipitation, but also elemental forms as adsorbents. We also have a possibility for biosorbents of the cells themselves. So there is a lot of possibilities for all these elements that we can use for, let me see if I can take this last. Yeah. So the reactor system, USB reactors, selenide and telluride formation, we show they are formed, but in not so high quantities, and to separate them then out as a biomass is a problem. Alginate beads, they work very good, but we have, we have the issue that they are expensive. And then the fungal systems, the nice thing is they work at low pH. So that gives then acid mine range has low pH, for instance. So fungal systems are very interesting for that. So with this, I come to the end of the presentation of the lecture. And so we have been working on several books. In 2000, we published the first book on treatment of sulfur pollution. And then in 2020, we have released the second edition, which is very also maybe interesting for you. Has been translated into, oh no, sorry, it's Portuguese, it's not Spanish, sorry, it's Portuguese and Chinese. And then we have worked on a volume for selenium removal, the rare earth pollution, and now we are finalizing the book volume on platinum group elements. So those books have with financial support of Science Foundation Ireland, they are all published open access, so you can find a lot of information in these books. So with this, I come to the end of the lecture, and I'm happy to answer any questions you may have.
Thank you, Professor Lenz, for your talk. So we open the question sessions. If you have a question, you can write it down in the chat here in Zoom, or you can use your microphone. And
yeah, you can also do it in English and Spanish, and it will be translated.
Okay, so we are open. If you have a question in Spanish, we can translate it to English. So, any questions? Professor Hugo Olvera has a question. Thank you for the presentation, Dr. Piet. I wonder if you have any thoughts about emerging bioelectrochemical systems for recovery of these kinds of elements. Bioelectrochemical.
Yeah, we worked on bioelectrochemical for instance, production of fine chemicals, so organics. But for the metals, we have not gone through that route. But there are other groups that have been working on that. And actually, there is also a scope for that. The nice thing is that with electrochemical, you can position the redox potential. So if for an element, you know that there it will be elemental form or a precipitated form, so you can steer the precipitate via the redox potential that you put on the electrode. So there is then the question is it a biological oxidation or reduction, or is it electrochemical, so without the microorganisms? But yeah, there are some very interesting works on that. And also, so I mentioned there, we can play around, for instance, the germanium, zinc, and gallium we can separate. We needed 3 reactors for that. Yeah, maybe with the bioelectrical systems, you would be also able to, within a reactor, also include one recovery process there. That's possible. But personally, we have no research on that. But if you're interested, I can point you towards some of this of the work of colleagues from, for instance, Título in Belgium. They have done a lot of work on that.
Thank you. Professor Hugo Olvera also mentions that he was part of the Ethicos 3 program back in 2014.
Yeah, of course. I still remember you, Hugo, no problem. It was
Hi,
actually
Prof. Piet. Very nice to see you. I was very happy when I heard that you were giving a lecture. It's very nice to see you.
Yeah, likewise. And also thank you for your mail message. I was going to answer after the talk, but yeah, it's incredible. It's already 10 years ago. Time flies. It's amazing.
Exactly, time flies.
Yeah, time flies.
Very nice to see you.
Yeah, likewise. Thank you for attending, and good to see that
Thanks for the talk.
Yeah, great to see your career also developing so well.
Thank you.
Thank you, Professor Olvera, for your intervention and your questions. In the auditorium, there is someone that wants to answer to do some questions to Professor Lenz. Well, while I have some questions, Professor Lenz, to your knowledge, is there what is the most, let's say, complete example of circular economy nowadays in the world that you know that has all the elements of this to fulfill the criteria of circular economy?
Yeah, very good question. I have to talk, but I think the most, I think the nice examples are the biorefineries. So where we take, for instance, agro-industrial residues, like as I mentioned, the brews and grains. But we can also consider even nuts, shells of nuts or rice. And we know the processes that can be recovering different fractions, also even grass in Ireland and in other regions. People are taking several components, chemicals out of grass, and on top of that, then the waste grass, it has also nutritional value. So I think there is a lot of, I don't know if you're familiar in Mexico with this technology readiness level. But at lower TRLs up to 3, 4, already quite a lot has been demonstrated. To really go then to full scale, then we get more issues with, for instance, legal aspects. If something comes from a waste, or if, for instance, if there has been pig manure somewhere, can you really go there for food or for feed again? So then we have regulatory aspects or the standards that play a role. We also have the acceptability. If we have a waste, we have in Ireland examples where they mix grass with the dung from the animal farm. Yeah, if you develop their chemicals, in principle, there is no issue there at all. But yeah, maybe public acceptance or even manufacturers might not be able to, or willing, they are able, but they are not willing to use those products.
Okay, thank you.
At the lower TRL, there are a lot of the agro-industrial residues. I think there we can already find some nice examples.
So long ago, some undergraduate students study the biorefinery process of extracting oil from grape seeds from the wine industry. So maybe do you know in Europe, a large wine industry, it's a common thing, or it's something that is implemented already?
No, I'm not aware that, but I'm not in. I don't have projects, or I don't work with the wine industry. So it might be they have that, but I'm not aware. But yeah, if it's done in Mexico, it should be also possible to transfer to other wine producing regions, Europe, France, but also South Africa, maybe also even Canada, US. So if there is a potential, but I'm not aware that this is a big. This is applied at a larger scales.
Okay, and another question more about the chemistry, about the precipitates. Have you done, or is it possible to do an X-ray diffraction experiments to know the crystal structure and maybe to study better the interactions to enhance the removal capacity of this?
I'm sorry,
for the
do
metals?
you mean to characterize the precipitates?
Yes.
Yeah. So typically I did not show that in the presentation, but typically we do the XRD for the chemical composition, but we can also consider other techniques, but they are mainly X-ray based for, so what is the metal speciation? At the time, we went also sometimes to Grenoble for xanes measurements, so for really speciation of the precipitates. We also look at settlability of the precipitates. Also, dewaterability is also a big factor that we can characterize. Zeta potential, so that is the charge that gives us an idea. Maybe by adding some compounds, we can increase the crystal size or the precipitation, that they precipitate better. So there are a lot of additional methods to characterize precipitates next to the efficiency. I think the two main ones are chemical composition, because that we need for maybe further processing. And the other one is this zeta potential, to have a crystal growth so that the crystals can grow slowly but larger so that they settle well.
Okay. Thank you. So we are open for more questions at the auditorium or here in Zoom. Okay. So if there is no further questions, Professor Lenz, we appreciate your time and your effort to give us this talk in the seminar, and we thank you very much.
Okay, with pleasure. Thank you so much for the invitation. It was a pleasure to share with you these data. If there are more questions, or if people want the papers or thesis of the PhD students, please send me an email. I can share those with you. There's no problem. Yeah.
Great. Thank you, Professor Lenz. And thank you all for your attendance, and see you
Yes,
next
thank
seminar.
you all for joining.